Yeah, my name is Mitch Nesbitt. I'm the Regional Sales Manager with Adama Canada, and I'm going to be your host for today's webinar. I really, first off, wanted to just thank everyone for joining today. We're certainly excited for the opportunity. Please use the chat function, as I mentioned. We will monitor the chat as the webinar progresses, and then we'll open it up at the end for any questions that you may have for the presenters. There'll be a QR code posted at the end of the webinar for those collecting CEU credits. So for those that have done these kind of events before, have your camera ready and your app open, and we'll try to leave it up long enough for everybody to gather their credits. Just a quick run through on the agenda today. So I'm going to introduce Corny Thiessen here shortly, who is our General Manager for Adama Canada. And then we're going to have Rob Barry, our Product Development Manager, give us a brief insight into launching and product development for Maxentis. And then our going to pass it over to Dave Greenshields, who's the president with Insight Plant Health, who's got some great lab work studies to talk to you about, and also give us an update on pulse disease management. And then we're going to pass it to Michaela Lobe, our technical sales agronomist for Alberta, to talk about Maxentis in the field. And then we'll wrap the day, like I said, with questions and any Q&A that you may have, and we will we'll finish things off there. So without further ado, I'm going to introduce Corny Thiessen. He's our Canadian General Manager. He manages the Canadian Adama business, and he's going to kick the day off with a bit of a welcome and introduction. So I'll pass it over to you, Corny.
Thanks, Mitch. And echo Mitch's comments, really appreciate you guys taking the time to join us here. I'm joining you from our head office in downtown Winnipeg. It's starting to get a little more mild now, so I'm sure we'll start to hear about those first few tractors or seeders rolling in southern Alberta any day now. Look forward to another great spring this year. I've been with Adama Canada for 4 years. I came to Adama Canada after working over 20 years with Monsanto and Bayer. And I came to Adama because there was some key trends I saw in the marketplace and some interesting opportunities that I thought those trends would reveal. Some of those key trends that I saw was the pressure of growing resistance and regulatory pressures, taking away from the toolbox that Canadian farmers have relied on to grow these diversified, profitable crop rotations. And in particular, that toolbox from traditional crop protection products was under pressure. We also saw, as a result of the consolidation and some of those regulatory pressures, that investment was moving more towards things like seed, biotech, digital, and biological platforms. Those are all great and necessary technologies, and I'm glad that there's more investment happening in those. But we still need synthetic crop protections to be successful in agriculture. And it is a concern that there isn't as much investment in that space as there has been traditionally. I think I was also saw a bit of a trend of decision-making moving out of Canada within some of the global companies. And I think that had a real risk of undermining that really inventive, innovative, entrepreneurial, and collaborative spirit that's made Canadian agriculture so successful over the last number of decades. Within Adama, what I saw that was appealing to me was a company that was 100% focused on synthetic crop protection products. With a long history of making more post-patent products, I also saw a growing R&D horsepower and a focus on formulation investment. There was a real inventiveness and ambition within the company, and also a great deal of autonomy granted to local businesses like Adama Canada. So combining those two things, the gaps that we saw in the marketplace with some of the underlying potential within Adama, it felt like a really great place to invent something new and build a business that could really contribute to Canadian agriculture, something that we are all really proud of today. So I see this event as a bit of a milestone for us in our evolution. We've worked hard over the last number of years to build up a team of great people that can help develop those next generation products and also can really partner with you in the marketplace to bring those successfully to market. We also are excited about the network of really smart people that we've been able to work with as we come up with new ideas, but then also develop those ideas and help to explain the potential value of them to the marketplace. And you'll see some of that here today. I'm a really big fan of that, that building networks of really smart people to look at things in new and different ways. I think that's one of the really great things about Canadian agriculture, is that we can still do that kind of thing. Lastly, I also use this as an opportunity to emphasize something that's really a key principle for us in how we approach the market. I think one of those great things about Canadian agriculture is that there's a history of everyone in the value chain respecting the roles of each other. I'm a big believer that suppliers and developers should be great at developing and supplying products, and that they should really trust and support distributors, retailers, advisors, and service deliveries to do their part well. When we all get together and collaborate, give each other feedback, call BS on each other, I think we all help advance Canadian agriculture. And we want a little bit of that spirit to come out of this event as well. Yes, we're excited about talking about our great new product. We hope it's a bit of a symbol for you of the evolution of ADAMA, both in terms of being able to be a good supplier of the more needed basic products, but also as a source of innovation for the future that can help continue that advancement in Canadian agriculture. And lastly, we hope that you see in this a sense of a company that's really committed to doing things the right way in the local market, and that we really truly have Canadian agriculture's interest at heart. So with that, I wish you a great webinar. Feel free to follow up and ask more questions. Again, call BS if anything here doesn't resonate with you, but mostly look forward to you interacting with some of the experts on the panel here. So good luck, and thanks very much again for joining us today.
Awesome. Thank you, Corny. And I've worked with Corny for a number of years. If anybody has a desire to even reach out and pick Corny's brain on anything, I know Corny is always available to answer some of those questions. So thanks, Corny, for that. I'm now going to pass, speaking of smart people, I'm now going to pass it over to Rob Berry. Rob is our development manager for ADAMA Canada. He graduated from the University of Manitoba with a Bachelor of Science in Agriculture in 1986 and a Master of Science degree in 1988. He has over 35 years experience in the ag industry, most of that in research and development with companies like Agribo, Monsanto, and ATP Nutrition. Rob joined ADAMA in 2015, has been leading our research and development program since that time. Rob, I'll pass it over to you.
Thanks, Mitch. Sorry about that. I couldn't get myself off of mute. I shared my screen, Mitch. Were my slides coming up?
You just got to take it out of presenter mode, and we should be good to go there. Yeah, if you
You can see them now.
Yeah, if you just start,
Okay.
it's still just in presenter mode, Rob.
There we go. All right, technology. Thanks very much, Mitch. So I'd like to spend about 10 minutes this morning just taking everybody through the development of Maxentis. It is a 6-year project from ideation to registration. The registration of Maxentis, we started with ideation, working with our global colleagues in 2017 to bring together the best active ingredients and formulation to provide growers with a truly elite performing fungicide. And I'll briefly take you through each of those steps, followed by some of our small plot results. So we tested numerous combinations of active ingredients for foliar disease control in cereals, sclerotinia control in canola, and disease control in peas and lentils. All were third-party independently contracted trials, so everything is done by third-party researchers. We found from our screening trials that 2 of the best actives to run with were azoxystrobin, a leading strobulin that's very strong on Ascochyta, Mycosphorella, and Anthracnose impulses, and prothioconazole, a leading triazole with excellent residual activity that's very strong on sclerotinia and white mold in canola, soys, dry beans, and other pulse crops. After the screening phase, we move into the prototyping phase where we develop numerous formulations and test them in the field. We started with a relatively simple suspension concentrate. An SC is a formulation that contains fine particles of the active ingredient suspended in water. Relatively easy to formulate, but they tend to have a shorter shelf life. Those fine particles can settle out over time, so you typically only have about a 2-year shelf life with a SC formulation relative to ECs or true solutions. And SCs often require an external adjuvant to enhance leaf penetration. We also developed some very good EC and OD formulations, or emulsifiable concentrates. An EC is essentially an oil-soluble active ingredient it's not particularly soluble in water, so you essentially dissolve it in an organic solvent to produce a liquid-like solution that's an emulsifiable concentrate. ECs tend to have enhanced leaf penetration relative to SCs, and you can do some novel, interesting things with ECs, like include a novel solvent system. They usually don't require an external adjuvant for that reason. And just from our prototype trials, we found that our EC formulations provided about 5% better efficacy than the SC formulation, so we moved to the SCs to bring to the market. In our registration trials, we continued to evaluate the different ECs and ODs that we had, and we found that the best EC with the Azorbitol 2 formulation technology provided about another 5% bump in efficacy compared to the other ECs. So we found over the course of a few years of testing our prototypes that we resulted in a 10% improvement in efficacy simply from formulation enhancements. It's amazing what the effect of a good formulation can do relative to something simple. We continued to fine-tune the rates, collected the final crop safety and efficacy data required for the key label registrations. Ultimately, over 100 trials were evaluated as part of our registration package that was submitted in early 2022. While we were waiting for the registration in 22 and 23, we continued to add to our knowledge base. We conducted more small-scale field trials to add to our label with more crops like corn coming down the pipe, blackleg and canola, and other pests for future label additions. We also initiated some lab and growth room trials that Dave will speak to here shortly. And very importantly, last year, we had over 50 farmer-applied large-scale field trials conducted prior to registration to ultimately affirm by the farmers that we have a winning formulation on our hands. And finally, in September of last year, registration was granted. Briefly take you through some high-level field results. So as I mentioned, we conducted dozens of trials over several years, focusing on canola for sclerotinia control, lentils and peas for ascochyta, mycosphaerella, and anthracnose control, and soybeans, dry beans for white mold control. And overall, Maxentis provided very good control across all of those diseases in our major crops, as well as overall a very good yield response as well. We also, in all of our trials, compare not just to the untreated control, but we compare it to leading standards like Lero in our pulse crops and Proline in canola. And in all cases, Maxentis performed very well relative to the leading standards. In addition to crop safety and efficacy data, we evaluate plant health benefits that may be apparent as well. When we talk about plant health, we're referring to the benefits of Maxentis over and above disease control. These can include enhanced photosynthesis, which can result in a greener crop until maturity and enhanced photosynthesis. Ultimately, the crop dries down, it doesn't die down due to disease. We can also get improved water use efficiency, and we can see reduced ethylene production by the plant. So the crop responds better to stress, such as heat stress, drought stress, and can respond better from the enhanced water use efficiency. So ultimately, that enhanced photosynthesis, improved water use efficiency, and better response to stress can result in higher yields, greater bushel weight, and better seed quality, even in the absence of disease. I'll take you through a few examples of those in our trials. We have numerous examples of these, but here's some particular trials that I'll show you. Last year in Elm Creek, we saw a good greening effect in a pea trial where we had close to 30% more green material close to maturity relative to the untreated check. And this means that the crop can photosynthesize optimally right until it's mature and that gives you the best opportunity for optimizing your yield. And again, Maxentis performed relatively well compared to other standards such as DX, Delero, and Quadris. We've also seen some numerous examples of yield enhancements in the absence of disease or very low disease pressure. Here's a couple of examples in lentils from southern Alberta the past 2 years, where we saw about a 15% yield bump from Maxentis in lentils when there was very little disease, again, from those plant health effects. And again, Maxentis performed well compared to leading standards like Delero and DX. We've also seen some good seed quality enhancements in our Maxentis trials. Here's an example from lentils in Minto last year, where we saw about 7% seed damage in the untreated control due to moldy seed, shrunken seed, where it dries out too quickly and doesn't get to be a nice big plump seed. It can be cracked. Many factors affect seed quality and cause seed damage. And Maxentis fungicide can certainly help to minimize seed damage. And here we saw a good statistical effect where we saw 3% less seed damage from Maxentis relative to the untreated control in lentils. We've also seen numerous examples of enhanced bushel weight, increased bushel weight. And here in our trial, one of our trials last year in Minto, in canola, we saw over a 10% bump in bushel weight from the application of Maxentis, again, helping to maintain that plump seed relative to seeds that are drying down quickly due to disease. So ultimately, Maxentis was extensively field tested since 2018. It's backed by over 100 third-party and farmer-applied trials. It provided exceptional disease control, yield enhancements, and crop health benefits in our research trials. We'll continue to expand the label crops and pests going forward, but we've seen that we've got a real winner here for canola and pulse growers for 2024 and forward. With that, I will turn it over to Dave Greenshields to talk about the lab tests that he conducted last year. Dave is the founder and president of Insight Plant Health. He has a bachelor's degree in agriculture and a PhD in plant pathology from the University of Saskatchewan, and worked in research positions at Riken in Japan, the Sainsbury Laboratory in England, and the National Research Council of Canada in Saskatoon. Before starting Insight Plant Health, Dave was the research director for the Saskatchewan Pulse Growers and worked in product development and business development at Novozymes BioAg. Dave has developed and patented seed treatments that peaked at over 50 million acres annually, huge number. And Dave started Insight in 2020 to help other ag companies develop and market their products with sound science. With that, I'll stop sharing and turn it over to Dave.
Great, thanks a lot, Rob. And yeah, thanks to Adama for giving me the opportunity to talk to everyone today. Hope that everyone can see this here.
Yep.
Good to go. Okay, so today I'm gonna walk through some results on testing Maxentis that we did here in Insight Plant Health. Before that, I'm gonna give a brief introduction to Insight, and then talk about pulse breeding. Just as my former role at SPG, I was intimately involved in some changes there. And it's kind of a hot topic these days in the Western Producer and on Twitter and stuff. So I thought I'd touch on that. And then go through diseases and disease management, and talk a little bit about fungicide resistance as well. So Insight Plant Health, we started operations in 2021, founded, incorporated in 2020, but actually pulled the pin on my other job and sat by myself in a little lab in 2021 January. So just over 3 years now. We're exclusively a business-to-business service lab, and we try to offer end-to-end custom lab and research services for different ag companies. We're based in Saskatoon with just over 2,500 square feet of lab space, and we have plant growth chambers, lab scale seed treaters. We have a spray chamber. We actually just got a walk-in cooler installed this morning. Walk-in growth chambers and a research greenhouse here on the premises as well. Oh, I should say too, these are the four of us here that work at Insight, Runa and I both have PhDs, and we kind of lead the science operations. And then Juan is a formulation chemist who does a lot of great work with microbial formulations. And then Cassidy McPherson works on project management and kind of the technical aspects of a lot of the work that I'll show today. So pulse crops in Western Canada, this is probably not a big surprise to anyone on the call, but they're about just over 7 million seeded acres last year. To pulse crops, lentils were the highest acreage. And in lentils, it's mostly reds, followed by large greens, small greens, and medium greens. Next up is peas, and it's mostly yellow peas, followed by green peas. Then chickpeas. Chickpeas vary wildly in acreage year to year. But last year, yeah, it was 315,000 acres and mostly kabulis, a few desis, and then dry beans and faba beans after that. So like I said, I want to talk a little bit about the pulse breeding landscape. So the breeding in Canada for pulses has been dominated really by public sector breeding. So Ag Canada and the CDC have, in lentils, the CDC I think has 100% of the varieties registered. In peas, there's a little more of a split between Ag Canada, the CDC, and then DL Seeds has some, and Lima Grain as well. Most of these varieties are either royalty-free through the CDC and the SAS Pulse agreement, or else they carry a small royalty on the sale of certified seeds. So that's kind of where some of the money comes back into the breeding programs. Farm safe seed is really common, so varieties last a really long time. And then breeding progress is pretty slow to show up in the field. And here's an example of that with yellow peas. If you look at the orange line, that's the 5-year average yield over on the right there. And then the blue bars are the number of acres for the different varieties. So the oldest variety is the most popular, CDC Meadow, and it's also the lowest yielding. So there is some opportunity to gain some yield by adopting some newer varieties. And so if you look at the pulse breeding targets from presentations from the CDC and Ag Canada, the first one is always with every breeding program, it's always yield, right? Yield. And then second is disease resistance, and that's kind of why it's an important thing to talk about, even when we're, the main presentation is on fungicides, but the other piece of that puzzle is breeding, or one of the other pieces. Rotation is also a huge part, especially in pulses. So disease resistance, you may have seen some announcements on a lot of money going towards root rot breeding efforts in peas and lentils. Mycosphaerella blight is also important. And in peas, powdery mildew, you have to have resistance to powdery mildew to register a variety in Canada. So that's always on the list. Lentils, Ascochyta, there's at least some resistance to Ascochyta in all of the main varieties. Anthracnose, there are 2 races. There's resistance to race 1, but not race 0. So that's a target. And then chickpeas, Ascochyta, and then the mysterious chickpea health issue that seems to keep popping up. I don't know how you breed for it if you don't know what it is, but I'm sure that's on the radar as well. And then herbicide resistance. So there are a couple new chickpea varieties that have solo resistance. And then lentils, most of the lentils have that IMI resistance as well. Then quality and various other traits come after that. So the SAS pulse growers, they represent pulse growers, and they're funded by a levy on the sale of all pulse crops in Saskatchewan. So they're by far the largest funder of pulse breeding in Canada, to the tune of around $5 million a year. Back when I was there, so it's probably gone up since then. Until 2020, they'd wholly funded the CDC pulse breeding program in exchange for commercialization rights to those varieties. But then in 2022, they announced a new breeding partnership with Lima Grain. So that breeding agreement with the CDC ended in 2020. And so they looked for other partners. And under this new agreement, then producers are going to have to pay a trailing royalty on farm safe seed. So there's a variety use agreement attached to the new varieties that will be coming out. And then some of that money will go back into breeding new varieties. So this hopefully will inject a little bit of competition and encourage new entrants into pulse breeding. So from that, I'll switch over to the diseases, pulse crops, and give a run through of a few of those. In lentils, I mentioned some during the breeding part, but the root rots are major importance. Ascochyta blight, anthracnose, botrytis, sclerotinia, and stemphylium. Of those, root rots and anthracnose are probably the most important. Peas, root rots again, and mycosphaerella blight. Sorry, guys.
All good. All good. Somebody's stealing the bandwidth over there. So you're you're good. No problem at all.
Okay. Let me know when you can see me again here.
Got it.
Yeah, I can see your slides. They're just in. Oh yeah, we're good to go.
There you go.
Okay. So yeah, I was talking about Phantomyces when I was so rudely interrupted by the internet cutting out. And yeah, the main problem with the Phantomyces is how long it lasts in the soil. So it starts out in the soil with these oospores. Here's a picture of one down at the bottom right. And these things can live for like 15 years. So, rotation helps because they don't all survive for 15 years. But, even if you stop for 15 years, start it again, there's a chance you could get a Phantomyces just from the soil. And so how these work is they germinate once they either get chemical signals from the host, from pulse roots, like lentil or pea roots, and then they release these little mobile zoospores that swim around in the water and infect roots, or else the oospores themselves, like the resting spores, can germinate and infect like that. They can infect any time during the season, and then they pretty quickly, once they infect, start making new oospores. So on the top right here is a picture of a lentil root from the field that was sent in, and you can see how many oospores are in there, and that's just like a tiny little portion. It's about as wide as a human hair, this root, and that's that's filled with spores. So, like I said, the main problem is how long they last. Here's a picture of a field that was split, and there were, I believe that on the right it was an 8-year break between peas. And then on the left, it was a 4-year break between peas. And you can see even a 4-year break wasn't enough to stop the root rots. So the key to identifying root rots is to dig up those roots. And usually you see in the field, it's a combination of a Phantomyces and Fusarium. If you infect plants in isolation, you can see differences between the roots. You can see these Phantomyces roots are darker than the Fusarium infected roots. But generally when we get samples into the lab, this is what they look like. It's just kind of water soaked, crappy looking roots. So you can use a PCR test to figure out whether you have the Phantomyces or not. The management of a Phantomyces basically comes down to rotation right now. Yeah, that's why it's a sad story, because it's yeah, it's just really not easy to control. There are a bunch of registered seed treatments for suppression of Aphanomyces. Ethoboxam is the main active, and then metalaxyl at a really high rate as well. But that, that suppression, because it can infect anytime during the season, yeah, it doesn't give a lot of advantage. Or I guess it doesn't give control, I should say. Yeah, I mean, at the top here, just saying that, when you're using farm safe seed, then it's always a good idea to seed treatment. So lentil anthracnose was first recognized in 1987 in Manitoba. Now most fields have at least some anthracnose. These ones don't survive as long. They survive for about 4 years in the soil. And then they initiate when there's some rain. Moisture allows the conidia to germinate on surfaces, forms these hyphae. First, it forms what are called primary hyphae that are big and they don't really enter the cells. And then it forms these secondary hyphae that enter the cells and just kill everything. So the disease usually shows up between 8-node and early flowering. And you get these lesions on the bottoms of the stems here. It kind of looks like a cigarette burn on the stems, and then they start to coalesce. Within those lesions, there's sporulation, and so the disease is what we call polycyclic. Many cycles can happen in a single season. So Mycosphaerella blight in peas, we'll talk about that. That can be caused by a complex of Ascochyta pinodes, Pinodella, and Pizzi. Pinodes is the sexual, or Mycosphaerella pinodes is the sexual stage of Ascochyta pinodes, and it's the most common cause in Western Canada. So you can either start out with infected seed, and that's why you should use a seed treatment. Infected crop debris is kind of the main culprit, and the spores will come off of the crop debris and then cause lesions on the leaves. And then again, it's polycyclic. So those lesions can produce new spores, and then those can kind of blow up in a field. So you'll get kind of a single point, and then it'll explode from there. Here's a nice picture of those lesions on the leaves. So usually they start to appear on lower leaves around the onset of flowering. And then if you get the stem infection, which I think Pinodella is the one that causes the most stem infection, it can just knock the plants right over and get a lot of lodging. And then, yeah, major yield loss when it's a wet year and you get an unchecked control. And then the last one I'll talk about is Ascochyta blight in chickpeas. I mean, very similar life cycle to the Mycosphaerella, but it's a lot more aggressive. So, in wet years, humid conditions, the infection cycles are so fast with this disease, like that polycyclic disease cycle. If you look at, here's a picture from the lab here where we got some samples. These are called pycnidia. They have the spores inside of them. And then this is a close-up of the pycnidia down here. Each one of these little dots coming out of here is a spore, and each one of those spores can cause a new infection. So, this probably has 100 pycnidia in this one lesion, and then each pycnidium has, however many million spores. That's why it goes so fast. Yeah, this is how you're going to earn your CCA credits with this, but this is the group 11 fungicide resistance, where the pathway that's blocked is. So in fungal mitochondria, the electron transport chain that produces energy, it prevents, these group 11 fungicides prevent electron transfer at the QO site right here. And all of the Ascochyta, chickpea Ascochyta in Saskatchewan, most of the lentil anthracnose is resistant to group 11. Also the Ascochyta Pinodella, which is part of the Mycosphaerella blight complex we tested with Adama, that we'll talk about a little bit later, is also resistant. But they remain the most common fungicides applied in pulses in Canada. So we did some work a few years ago with Bayer to screen a bunch of isolates that they had collected and see whether they were resistant, and then sequence the gene that makes this cytochrome B complex in the plasma membrane. So we sequenced that cytochrome B gene in all of these different isolates and found that, so if you look at the graph, the leftmost green bar is a no fungicide control, and then the three right bars are different levels of pyraclostrobin or trifloxystrobin. You can see these first three are controlled by those group 11s, but then the rest of them weren't. And then all of the isolates that were not controlled by the group 11s had this G143A mutation in that cytochrome B gene. So with that information, we figured out an assay where we can take a small piece of a lesion and put it in a tube and then run a PCR on it. Same kind of PCR that they did with COVID, but this is for lentil anthracnose. And then use the mutation to cause a shift in the size of the fragment that's being PCR'd. So we get samples into the lab. Turnaround's usually about 50% samples, and we can tell you whether you have resistance or not. But looking at these numbers here, so this graph shows the percent of resistant isolates that we've tested in every year, 2021, 2022, 2023. And yeah, so most of them are resistant. So if you think you have resistance to group 11 in lentil anthracnose, you do. But I mean, part of that is selection pressure as well, right? Because these were almost all treated with a group 11 fungicide. So whatever's left over is probably resistant. Okay, now I'm going to switch over and talk about the lab and greenhouse testing of Maxentis that we did with Adama. So we did two different assays. The first assay was kind of a screening assay where we looked at, in Petri dishes, we looked at fungal growth and germination inhibition. We looked at 15 different fungicides. So these were competitor fungicides, Maxentis, and different formulations, and at different rates to generate 44 different treatments, and tested those treatments against lentil anthracnose, mycosphaerella blight peas, faba bean ascochyta blight, and then sclerotinia from canola. I guess the take home is Maxentis inhibited the growth and germination of all of these pathogens. So I'm not going to throw up, like a bunch of Petri dish pictures. I will throw up some Petri dish pictures, but if they were just the Maxentis ones, they're just empty Petri dishes. So it worked quite well in that test. The other testing we did was fungicide migration testing, where we tested 9 different fungicides for their ability to control sclerotinia in canola, and then 11 different fungicides for their ability to control mycosphaerella blight in peas and anthracnose in lentils. And I'll describe how we did the migration here shortly. And Maxentis in this trial was among the top 4 fungicides in each of these, controlling each of these diseases. So here's kind of here's an example of the plate assay. So these are just Petri dishes at different rates of Soratel. Here are the first 3 rows, and then Proline on the bottom row, or on the second to the bottom row, and then the control with no fungicide on the bottom row. And then this first panel is Ascochyta pinodella, part of the mycosphaerella blight complex, and then Ascochyta fabae, which is fabamine ascochyta on the right side. And kind of the take-home message here is that formulation matters, right? Because you look at the Soratel, it's the same rate of prothioconazole as this one here, but you get some growth from the Proline, and then you don't, even if you go to a lower rate, there's no growth of the Pinodella. There's a little bit of growth here over on the fabae, but at the same rate, it's controlled. So I'll show a few more of these kind of examples, but that's just how we set it up. So basically, you take the fungicide as it would be tank mix. So if you had a spray tank and you just added agar to it, that's what would be in this this Petri dish. So for the migration testing, the way we set that up is we took wax paper and then wrapped it around half of the leaf, one half of the leaves that were being sprayed. And then we put those in the spray chamber here, and the spray passes over top, and it covers the half leaf that's exposed, but not the leaf, not the part that's under the wax paper. So if you go into a close-up here, you can see that half of the leaf gets fungicide and the other half does not. Then we cut those leaves off and we inoculate them. In this case, Cassidy is inoculating soybeans. What I'm going to talk about today is with canola, peas, and lentils, but it's the same assay basically. So you spray half the leaf, and then you inoculate the half that was not sprayed, and then you look to see how long it takes for the fungicide to move from one side of the leaf to the other and show control on that side, and then also how long it takes for it just to disappear and there's no control. So this is that assay with Sclerotinia. The rows here this is the sprayed side, so it's kind of the control. This is 1 day after spraying, 4 days, 8 days, and 12 days after spraying. And then the columns, the first column is the water control, so it was just sprayed with water. Then Soratel, Proline, Quadris, Maxentis, Proline Gold, and Cotegra. And so what you can see here is the red outlined boxes are ones that had complete control. And so after 8 days, Soratel and Maxentis showed complete control in all of the leaves that were inoculated. So if you had parts of the plant that weren't sprayed, within 8 days, those parts would have enough fungicide to control the disease. That was the same for Cotegra. Proline Gold showed up in 4 days and then lasted to 8 days. But by 12 days, the control wasn't there. The Soratel, Maxentis, and Cotegra all controlled out to 12 days. And we didn't test past 12 days, so we don't know really how long. But with the new assay that we're running soybeans that I showed before, we're going to try and test a little longer. So this is a measurement of the lesion sizes. So, the visual showed some of the results, but if you look at 8 days after spraying, Soratel and Proline Gold showed complete control, and then Maxentis came in third there. And then 12 days, Maxentis and Soratel continued to show strong control. And then here are the plate assays with Sclerotinia. This is just showing kind of the actives that are in both of the Soratel and Maxentis, but in other products here. So the Proline at the top showing control. The Quadris did not control this Sclerotinia in this assay. Soratel, there's a lot of different rates of Soratel here. It was controlled at all rates. And Maxentis, which has both of those actives, also controlled at all rates. So looking at peas, this is peas 8 days after spraying. The sprayed side here is on the left column, and then there's the control, the water control at the top here. Soratel, Proline, Quadris, Maxentis, Delero, Cotegra, Miravis Neo, and Elatis were the ones that we tested for peas. And then, yeah, if you look at the Maxentis, which is kind of our focus here, control on all the leaves. So there was no lesion spreading on those, again, 8 days after spraying. So if you look at the same graphs, Soratel, Maxentis, Miravis Neo, and Elatis all showed comparable levels of control. 8 days after spraying, 12 days after spraying, again, Soratel and Maxentis showed strong control. So the control lasted longer. It came on about the same amount of time, but lasted longer than the other ones. Then we combined all the different time points here to show, yeah, like, so we took all the data from day 1, 4, 8, and 12 and combined it, and then just looked at the lesion size overall. And then the best performing fungicides, when you're looking at all of them, were Elatis, Soratel, Miravis Neo, and Maxentis EC. This is again showing the inhibition, and again it's another kind of formulation matters slide, right? So this is Vascucita pinedella. Looking at Proline, you do get some growth on the plate. Obviously, there's, I mean, quite a bit of suppression compared to the control, but there's still a little bit of growth. Quadris, there's quite a bit of growth there. Not much control. Soratel, no growth, even at the lower rate, there's no growth, but at the comparable rate, it's complete control. And then Maxentis, not surprisingly, also complete control. Oops. Okay, so we look at Ascochyta and faba beans. The Proline, quite a bit of growth. Quadris did a good job here, actually, and controlled. Soratel, there was control, and then Maxentis EC, complete control again. So lentils. This one is kind of hard to see, and it was even hard to judge. You had to look at each individual leaflet on the leaves, and then count the number of leaflets that had lesions on them. So it's kind of an unpleasant test. I don't know if I want to do that one again, but this is the water control here at the top. Soratel, Proline, Quadris, Maxentis, Elatis, Miravis Neo, Cotegra, and Delero. And then again, if you look at Maxentis, good control. There's maybe one lesion. Can't tell if that is at the top there, but if we look at the graphs in the next one, oh, actually this slide here is showing just the difference between the different isolates we used. So we used an isolate that's resistant to group 11s. And so this gives you a visual. This is an isolate that is not resistant to group 11. So if you put Quadris on a petri dish and you put a bunch of spores on the petri dish, the spores don't germinate. This is just a water compared to Quadris. And then down here, this is a resistant isolate of the lentil anthracnose, and you can see the spores do germinate when you put the Quadris in. Not as much as they do on the control, but still quite a bit of germination. So, and that's the same isolate that we used for this lentil testing. So for the lentils, the migration showed up sooner. So 4 days after spraying was when it really showed up. It showed up 2 days, or 1 day there was a little bit, and then 4 days there you could see control in Soratel, Proline, Maxentis, Cotegra, and Elatus. But then by 8 days that had mostly just gone away. So this is the check on the one part. I mean, there's a little bit of variation between the different treatments, but most of the control was gone. So here's another, just on the Petri dishes, this Quadris, kind of what I showed before. I mean, the isolate's resistant to group 11, so it's not surprising there's growth on there. There was a little bit of growth as well on the Proline, which is kind of surprising. But again, Soratel showed no growth on any of the different concentrations. And then Maxentis, again, no growth. So for conclusions part 1, I guess, pulse acre
Just like a TV series, left us on a bit of a cliffhanger.
Michaela, maybe what we'll do is we'll let Dave, he could jump back in after and share his conclusions, but maybe good spot to jump over to you. So next up on the agenda, we'll let Dave jump in after is Michaela Lobe. So Michaela's our technical sales agronomist for the Alberta region. She has a Bachelor of Science degree in agriculture and carries her CCA and PAG designation. She's held a variety of roles in her career, including field research, agronomy, and sales. And she joined us in the spring of 2023 as part of our growing technical sales agronomist team. So we've learned from Rob getting products developed. We've just heard from Dave in terms of some great lab results and just a lot of interesting work that's being done. Now, how did Maxentis fare in the field? So Michaela, I'll pass it over to you.
Okie dokie. Can you guys see my presentation? Or are you seeing presenter mode?
You're in good shape.
Okie dokie. And then Dave, we will jump back to you at the end. We just thought, just in case you were going to take a little bit longer to get back to where you are. Like Mitch said, my name is Michaela Lobe. I'm the technical sales agronomist for ADAMA in Alberta. I started here last March, and I promise my presentation will be better than my jokes, so we'll dive into it. For those of you who don't know, the TSA role is new at ADAMA. They started this role in 2023, and it's just another way to show that ADAMA is growing, ADAMA is investing, and trying to find the best solutions for Canadians and Canadian agriculture. Our main role is supporting the ABMs, which is in turn supporting you guys if you have any questions or any concerns. And then we're also in charge of running all of our research authorization and our multi-product comparison trials. So some of you may have been out to those this past season where we're just trying to showcase our pipeline so that you guys are prepared and know what's coming from ADAMA in the future. So to get Maxentis ready and launched for 2024, we partnered with a company called Intent. They are a third-party company, and we use them because they are focused on field-scale farmer-run trials and data collection. This was advantageous for us as they have a large network of growers, and we wanted to make sure that we were testing Maxentis throughout all the prairie provinces to see how it was going to perform in different areas under different growing conditions. This was done by having over 50 trials throughout Western Canada, also some in Eastern Canada. Here is a list of all the trials that were done in Western Canada. If you see one on here and you want the specific data from it, don't hesitate to reach out to any of your TSAs or the ABMs or anyone within ADAMA, and we will get that information to you. I'm not going to go over too many specifics in this presentation, just a bit of a broad overview. This is how our research authorizations are set up. For those of you who don't know, a research authorization is regulated through the PMRA, and it's the last step before a product gets registered. It's large scale, it's farmer run, just so we're able to get this product and understand how this product is going to work before we register it and sell it to farmers. So we had set this up for all 3 crops against the major competitors on the market. We wanted to see how we were going to stack up against them. We didn't want to hinder ourselves and make sure that we were performing to the best that we thought we were going to perform. So just to note, we were testing 2 different rates of Maxentis in our pulses. So in peas, we were testing Maxentis against Delero and Miravis in an untreated check. In canola, we were testing Maxentis against Proline, Proline Gold, and Soratel. And then in lentils, we were testing the 2 rates of Maxentis against Delero and Elatis. All of these were against an untreated check or grower standard practice, depending on the growing conditions and disease pressure. Now I'm just going to quickly touch on or go over, this is just the generalized summary of the yield data. So this is all the pea trials throughout Western Canada. As you can see, there was a little bit of a yield difference between our untreated and our treated. Honestly, we probably would have expected to see this a little bit higher. I think that just has to do with the growing conditions and when disease was present when we were spraying fungicides this year in the pulses. There was a little bit more of a difference here in lentils, but as you can see, Maxentis was right up there with them, and then actually in lentils, the high rate of Maxentis, which is the rate that we registered, the 422 mil per acre rate, did win the majority of the trials. This is probably the most interesting of all the crops. Canola is usually that one crop that gets forgotten about. People don't think it pays to spray. They don't see a disease pressure usually, or they don't think that it's worth it. This year, it was interesting to see in all these yield trials that we did, there was on average a 7 bushel increase between our untreated and our Maxentis, which is huge. Clearly, it paid to spray, even on a year when it was fungicide timing. Might have not really looked like it was a fungicide year, but as we can see through all this yield data, it definitely was. I'm going to dive into 3 here. These maps here are really cool to look at. This is one of the beauties of working with Intent. They were able to give us these vegetation maps where we're able actually to paint a really good picture, and going into harvest, we were able to see how well it performed. So these maps were taken late August. You can see throughout all my ratings, I didn't see a huge difference when looking at the plant stand between all of them. But as you can see, those 2 very distinct red lines there, those are the 2 untreated checks that were in this field. And this grower, if he had his whole field yielding 75 bushels, which the untreated check did yield, he probably would have been very happy. However, he was tickled pink when he saw all of his treated acres. The Maxentis went just shy of 84 bushels, and the rest of the products were also in those low 80s. So that's huge. That's about a 9 bushel increase. So definitely paid to spray here. One thing to note, a lot of the real visual maps that we saw were in areas where there was a higher disease pressure, a little more moisture and things like that. This one was just a little bit farther south in Alberta, just outside of Lacombe. As you can see, the treated was on the north side of the field, and then there was a clear, distinct line in the middle there. That again was our untreated check, and there was a 29% yield increase between our untreated check and Maxentis. And again, that is huge when you're thinking on a canola, how much that would work out to in your ROI. And now we're going to go a little bit to the east. This was one out in Manitoba on peas. Again, these visuals you can see are very distinct. On the right side of the field here, we have that a lot more red, a lot less green biomass. And the untreated check, which is the right side of the field here, was yielding about 61. Again, not a bad yield considering the year. But on the right side or the left side of the field, sorry, the Maxentis yielded just shy of 78 bushels. So that's a 16 bushel increase. And what is allowing Maxentis to be a leader in the fungicide market already? As we have talked about, or you may have heard, it's our formulation. This is the first time you're seeing these 2 active ingredients mixed together, prothioconazole and azoxystrobin. That paired with our azorbital formulation that is in this, azorbital 2, is a unique solvent system that is allowing for faster and efficient absorption of this product. And by getting it into the plant quicker, we are getting excellent control and coverage because of its systemic movement within that plant. And through David's presentation, you will have seen our active ingredients and the way they are formulated are able to stay in that plant a little bit longer, just giving you a little bit more control throughout that growing season. And here are the meat and the potatoes of Maxentis. As you can see here, this is just all the technical information. It is prothioconazole and azoxystrobin mixed together. It is an EC formulation, and by being an EC formulation, it's easier handleability, and also it has smaller particle sizes, which are allowing it to get into that plant quicker, which is super beneficial when you're working with something like prothioconazole. Prothioconazole is very sensitive to photodegradation. So if it's sitting on that leaf surface and not actually getting absorbed into that plant quickly, it is susceptible to being broken down by sunlight. So once we're able to get that plant and that active ingredient into the plant quickly, it's able to work that much more effectively because you are getting it more product into the plant for better control. If you are curious about packaging sizing, here is what it looks like for Maxentis. It's a 20 acre a jug rate for your pulses and 19 acres for your canola. Call it 20. And it is also available in bulk, which is nice with this being an all-in-one formulation. If you are looking at what your pulse products or what your pulse options might be for Adama products, depending on when you have the onset of disease, if you have disease coming in very early, you might consider going with something like a Top Notch, followed up with another product later for those white mold or later season diseases. Or if you're looking for a one-stop shop, Maxentis is an excellent option. Now I'm just going to quickly touch on a couple of the other fungicides that Adama has because I think they are a great fit for the market. Sora Duo is our prothioconazole tebuconazole combination. It is also powered by azorbitol, so you are seeing it being able to get into the plant quicker and have excellent control. One thing to note, we are working on a Sora Duo label recommendation amendment, so that there will be news to that to come. But one thing to note, we're looking at extending that label to changing the rate a little bit so that we could have an option for flag leaf timing. And then, which you guys have seen a lot throughout David's presentation, but Soratel. So this is our prothioconazole. And one thing to note, what stands us apart from some of the major competitors on the prothioconazole market, we do have it in an EC formulation compared to an SC formulation. And that, like everything that I've laid out already, but that paired with the azorbitol that is in it is super beneficial. It will increase your plant's uptake and penetration, which in turn, by getting it into the plant quicker, you're going to have reduced runoff, reduced photodegradation, and then improved rain fastness because it's able to get into that plant and have better efficacy once it's in that plant and working. That's all I have for my presentation, so I will pass it back to everyone else.
Perfect. Thank you, Michaela. So Dave, we'll turn it over to you to just wrap your piece up there, and then we'll get close to wrapping this up and open it up for questions.
Okay, sorry about that, guys. This never happens to me. But yeah, so I was just going through the conclusions, I think, when I cut out. And the last conclusion, I mean, the conclusion on the Aphanomyces is that it's still it's the single biggest threat to PM lentil production in Canada. And rotation is kind of your main tool there, combined with the seed treatments. So with all of the other diseases that we talked about, though, foliar fungicides are really the important tools that you need. The conclusions number 2, so the conclusions around the testing for Maxentis, formulation matters. And Michaela was just talking about that, and it does. Like in these trials, yeah, it really showed that you can have the same active formulated different ways and you get different results. And you get different results both in terms of the level of control, just, you know, in a Petri dish assay, but you also get different results in terms of how long it takes for the active to migrate, and then how long it lasts in the plant. So Another one is that Maxentis EC controls group 11 insensitive fungi. It controlled all of the fungi in these assays. The migration in canola and peas took around 4 to 8 days. So if you had, unsprayed parts of plants, 4 to 8 days later, those should have control as the active would have moved down. And then the timing is shorter in lentils. So it takes 1 to 4 days, and then it doesn't last quite as long. Both Soratel and Maxentis provided control up to 12 days in canola and peas, but only around 4 to 8 days in lentils. And they were, Soratel and Maxentis were among the top 4 treatments in all of the crops and diseases tested. So, yeah, so I mean, it was fun work to do, and it's, it's always kind of satisfying when you're working for a client and the product actually works, and you can tell them the good news rather than breaking the bad news. So, yeah, it was a fun project, and hopefully my technical issues didn't take away too much from what we did, and it was clear. So thank you again to Adama for giving me the chance to share the results.
Excellent. Thanks, Dave. We will, if you're willing to stick around, we'll open it up to questions here shortly. So did want to just reiterate a couple of key takeaways when I listened to Rob and Dave and Michaela, and if I was going to kind of put it into a 3-sentence synopsis, would be what Maxentis, registering a product like Maxentis takes considerable amount of time to get to where we are today. And it's a culmination of 6 years of work, identifying the right actives, the right formulation, and the best system to bring that product to market. And we heard it time and time again today that formulation matters. And I think in Dave's work is really interesting when you have the same active ingredient at different rates, how that shows up in not only the Petri dishes, but some of those migration tests as well. And Maxentis is an advanced EC formulation that was the right choice, even as it moved through Rob's pipeline. And if you leave you with one thing today, it's that Maxentis will deliver industry-leading performance on key diseases in canola, pulses, and soybeans. Feels like we've been able to prove it in the lab, and even more importantly is when we put it at field scale replicated trials that we mirrored that performance. So hopefully that came across in the presentation today. And just in case you got to hear from Dave, if you're interested in reaching out, I did want to just say thanks. We mentioned it at the start that we work with third-party research companies to provide unbiased data and unbiased results so that we know exactly what we're looking at. So if you're interested in reaching out to Dave or giving him a call, phone number's there. The email is info@insightplanthealth.com. You can be found on the web at insightplanthealth.com as well. So thank you to Insight and Dave for joining today and sharing the story from this last year. Then as mentioned Intent, which is our third-party field scale trial company that we used this last year. You can see the information there as well if it's something that you're interested in or something that you're looking forward to trying to work with in your day-to-day operation. So thank you guys very much for that. So we're going to open it up for any questions. And before I do that, I just wanted to say thanks to Michaela, thanks to Rob, and thanks to everybody for attending today. We had a strong contingent of folks on the call. I just want to say thanks for taking time out of your day to join us today. If you've got any questions, please reach out to your area business manager, reach out to myself, your technical sales agronomist, or even right to Courtney Thiessen, our Canadian lead. The door is always open, and we'd love to hear from you. So with that, I'm going to open it up for questions. You should be able to see the QR code on the screen for your CEU credits. And now if somebody wants to help monitor the chat, we'll open it up for questions.
Maybe I can start, Mitch. I saw one earlier about Soratel versus Proline. Why did it perform better in the Petri dishes? Great question. And honestly, going into it, I didn't think there'd be a difference in formulation on the Petri dishes. We've seen the formulation benefits in plants, in the migration tests, in European studies, and it was repeated in Dave's lab. The spores was really interesting. So it's really interesting. We're not sure why, but obviously there's a formulation advantage in our sorbitol technology with the EC formulation that's even having a positive effect on controlling spores or sporulation relative to an SC formulation. Can't say why, but obviously there's a formulation benefit on this on the spore control there.
Excellent. There was another one here. Clear benefits to both Soratel and Maxentis, but when should you choose Soratel and when should you choose Maxentis? This is a great question. They're definitely both have a fit. They're both going to do an excellent job. In my opinion, if you have really high disease pressure or you're someone who thinks that they might have to do 2 applications of a white mold or a sclerotinia fungicide, I would go in first with a product like Maxentis and then go in with a straight prothioconazole second. So I would do a Maxentis first and a Soratel second if you were doing 2 apps. Or if you just have really high disease pressure and you think that it is a disease year, I would go with the premium product of Maxentis.
Yeah. And to add to that, in canola, both Soratel and Maxentis are great choices, very good control that we've seen. There could be a little bit of a benefit on the plant health effect from the strobe in particular in canola. Efficacy-wise, they both perform very well in canola, as we've seen.
We've got a non-agronomic question. What is the MSRP of Maxentis? So good question. So to be 100% upfront, we don't publish an SRP, so it's a bad default. But talk to your local retail that you work with, and they'll be happy to share. But similar to the rest of our products, obviously our strategy is to make it right priced, and we don't have grower programming, so it's a direct price to the grower, and I think good value there. And I think when you get into some of those conversations with your retailer, you'd be pretty happy with where the product is lining up. It's a political way of addressing that. Any other questions?
The question on availability for this season.
Yeah, great question. So we have a full production plan, and we've taken an aggressive direction on supply. So once again, we are doing a full launch, but it is one of those things that, as we expect, strong demand for the product. To talk to your retail, there are some things happening even now over the next couple of weeks in terms of some deadlines. So if it's something that you're interested in, always good to be proactive and reach out to your retail. We lean on them for supply, but certainly we would expect significant volume to be available.
And one thing to note, if anyone is having troubles with the CEU or the QR code, just you can email us and we can send you either a copy of it or the physical sign up sheet, and you can sign yourself in and we could submit it from there.
Okay, if there are any other questions that come up after the call today, just please forward them on to anyone. You could even reach out to Dave if anything regarding the work that he did, or like we mentioned, your area business manager. So we'll be happy to address any of those even after the call and could send out a follow-up. So just wanted to finish by saying thanks again to the presenters today. Thank you to all the guests. We had at one point about 240 people on the call today. So thank you very much for your attendance. If you've got any issues with the QR code, we'll lean on Michaela to deliver that. And just wish you a great season this year. And thanks for the support for those that are working with Adama today. And if you're not, hopefully products like Maxentis and some of the other exciting things we have coming down, hopefully we'll do another one of these in the near future with one of our other interesting solutions that we're hoping to bring to the Canadian marketplace. So thank you for your time. Thanks for the day, and be safe out there.